Welcome to GammaLab. This is the home screen, the starting point of the platform. At the top, we see the 5 main sections of the system. Home, Preprocessing, Analysis, Measures, and FAQ. On the left side, the selected signal panel appears, where the active signal is chosen for all further operations. Let's begin by loading a signal. I click Open Signal. This plugin allows loading EEG signals in ABF or EDF formats. The upper Open Signal button opens the plugin, while the lower Open Signal button opens the File Explorer. After selecting the file, the signal is displayed in the central workspace. If the signal has more than 3 channels, only the first 3 are shown. On the right panel, all channels are listed. I can enable or disable each one to control which channels are visible. I now zoom in, move across the signal, and interact with specific points for viewing. If I want to open a new signal, I return to Open Signal, load the file, and then choose the active one in the selected signal panel on the left, for viewing. Preprocessing. Now I open the Trials plugin. This tool segments the continuous signal into trials around detected events. In Channel, I select CA1, the channel used for extracting the trial data. In Stim channel, I select IN7, the channel where stimulus events occur. Threshold is set to 0.700, meaning an event is detected each time the stimulus surpasses this value. Stim number is set to 1, because we expect 1 stimulus per trial. In the Time section, I set the initial time to -0.005 seconds and the final time to 4 seconds. In Trial End mode, I select Cut to the next stim, meaning the trial ends when the next stimulus is detected. I press Generate Trials, and the trials appear. I can move through them using Previous and Next or discard them with Discard. Next, I open the Remove Artifact plugin. This plugin is used to correct artifacts by applying a smoothing or interpolation method to a selected interval. In mode, I select Cut from Start. First, I set a cut from the start at approximately -0.079 seconds. Then I select Interpolate Interval, define point A at 0.053 seconds, and point B at 0.0832 seconds. By pressing Remove Artifact, the system replaces that interval with an interpolated version, ensuring signal continuity. Now I open the Filter plugin. This plugin applies frequency filters such as high pass, low pass, band pass, and notch filters using a Butterworth design. I interact with the graph, zoom, and inspect how the filtered signal behaves. Adjusting the frequency range and filter order allows removing noise or isolating specific frequency bands. Analysis. Time, Frequency Domain. Wavelet. I now open the Wavelet plugin, which computes the continuous Morlet wavelet transform to display a time, frequency representation of the first trial. Sample density defines how many samples per unit of time are used for the transform. I set it to 1000. Low frequency and high frequency define the minimum and maximum frequencies shown in the scalegram. I set them to 1 and 500 Hz. Cycles determines the number of oscillations in the Morlet wavelet, controlling the balance between time and frequency resolution. I set it to 2. I press Create Wavelet and the scalegram appears. This button performs the actual wavelet transform and generates the visualization. I enable logarithmic scaling using Scale, which changes the frequency axis to log scale. I also switch normalization to Z-score, which standardizes the power values to improve visibility across frequencies. Wavelet Average. Next, I open Wavelet Average. This plugin computes the wavelet transform for all trials and averages the results, highlighting consistent oscillatory patterns across the dataset. I use the same parameters, sample density, frequency range, and cycles. I press Create Wavelet. The button performs the transform for every trial and then computes the average. In the bottom left corner, the progress indicator shows the transformation process. I activate logarithmic scale again, apply Z-score normalization, and interact with the averaged scalegram. Time Domain. Average. I now open the Average plugin. This tool computes the average of all active trials to produce a single clean signal that reduces noise and enhances event-related responses. The button Calculate Average performs the averaging and displays the result. I interact with the curve and inspect its structure. Event-related power, ERP. Next, I open event-related power. Pressing plot ERP produces two synchronized views. The butterfly plot, showing all trials superimposed. The heat map, showing trial power over time. The list panel allows enabling or disabling individual trials. The range panel restricts the trials shown to a specific numerical interval. I modify the list of active trials, switch to a single trial, and adjust the trial range to observe changes. This allows me to inspect both global and individual trial behavior. Frequency domain. FFT. The FFT plugin computes the fast Fourier transform to show the amplitude spectrum of the signal. Sample density sets the number of samples used in the transform. I set it to 1000. The low and high fields in range define the minimum and maximum frequencies displayed. I choose 0 to 500 Hz. After pressing Calculate FFT, the spectrum appears. I interact with the plot to examine dominant peaks. FFT average. With FFT average, the FFT of each trial is computed and then averaged, reducing noise and producing a more stable spectrum. The fields have the same functions as in FFT, sample density and frequency range. I press Calculate FFT average and explore the averaged frequency response. PSD. The PSD plugin calculates the power spectral density using the Welch method. Calculation mode lets me choose whether to compute the PSD for all trials, the average, or a specific trial index. Then I configure the Welch parameters. Window, the tapering function applied to each segment. N per seg, the number of samples per Welch segment. N overlap, overlap between consecutive segments. NFFT, the length of the FFT used in the calculation. The range panel defines the frequency interval displayed. I keep the trend set to none. After pressing Calculate PSD, the system displays how power is distributed across frequencies. PSD average. The PSD average plugin computes the Welch PSD of the average trial signal, producing a smooth view of overall spectral characteristics. I configure the sample density, the Welch parameters, and the frequency range. I press Calculate PSD average and interact with the averaged PSD plot. Relative PSD. Finally, I open Relative PSD, which computes absolute power in a selected frequency band. Relative power as a percentage of total power. Sample density, Welch parameters, and the trend work exactly like in PSD. In range, I configure the band to 8 to 12 Hz. I press Calculate Relative PSD. The results appear in the absolute power and relative power fields. Measures. Zoom tools. By right-clicking any graph, I open the contextual menu. Horizontal zoom magnifies only the time axis. Vertical zoom magnifies only the amplitude axis. Both axes zooms in on both axes simultaneously. Reset view restores the original full view frame. These tools help analyze fine details in the signal. Slope measurement. Right-click and select Slope, 2 points. After clicking the first point and then the second, a dashed blue line appears, representing the slope segment. In slope results, the table displays delta x, delta y, the slope value, the segment distance, and the timestamp. Amplitude measurement. Next, I choose amplitude. Right-click and select 2 points. After selecting the 2 points, a message shows the amplitude details, and a red dashed line appears. In amplitude results, I see peak-to-peak amplitude, minimum and maximum values, their positions, the window boundaries, and the number of samples inside the window. Slope for all trials. Right-click and select All Trials, using this slope to applies the slope measurement to every active trial. In the Measures panel, the table updates with the measurement for each trial. Export CSV. Using the Export as CSV button, I download all measurement data. I open the exported CSV file in Excel to show the structured results. Export images and tables. By right-clicking any graph, I can choose Export as image, saving the graph as PNG, JPG, BMP, or TIFF. Export as table, exporting the underlying data as CSV, XLSX, or JSON. These options allow easy integration of results into reports. FAQ. In the FAQ section, I can access all official project resources. The user manual. The official GitHub repository. The complete project documentation. The tutorial video playlist. And the link to download the latest version of GammaLab. This section serves as a support center and extended documentation for users. This has been an explanatory video showcasing the main functions of GammaLab, the open-source software developed as the graduation project of students Sergio Asencio Rodriguez, Matías Felipe González Valencia, Jorge Esteban Martínez Clavijo, and Giseth Villalobos Rocha at the Pontificia Universidad Javeriana. Long live free science!